Pensez-vous que nous attendons encore des personnes? Yes. Yeah. So I'm going to start, and I'm going to start in English because most of the committee members need to understand what I'm going to say. So today we are here for Edel's PhD defense, and I'm going to introduce the members of the committee. Starting with myself, because I will be chairing this committee. So we have one of the members is following us, remotely. So yes, I think he can see me. So I'm Emmanuel Cam. I'm a professor at the University of Western Brittany. And I'm not working on fish, not even on salmon. I'm working on seabirds. So I've probably been invited because of my position, professor in the university, woman, and also the fact that I'm working with market capture modeling, because we need a university professor in the committee to deliver a diploma. So we also have, okay, I hope things are going to work. We have a reviewer for Edel's thesis who's with us using a video conference. I apologize for the French pronunciation of your name. So it's Samu Mantiniemi. So,
well, very well, thank you. Yeah. Good day to all. My name is Samu Mäntyniemi. It was very well pronounced. I'm from Finland. At the moment, I work for Natural Resources Institute Finland, which is a governmental research organization. My background is in Bayesian statistics and market capture modeling. At the moment, I work with estimation of population sizes of large carnivores like bears and wolves. But in the past, I've been a lot of involved in salmon research as well, and I have done a lot of similar work that will be presented by Edel today.
Okay, thank you very much. So it happens that I'm also a reviewer. So I'm the second reviewer for this committee. And then we have two examiners. So we have Ian Malcolm coming from Scotland.
Yep, so my name's Ian Malcolm. I'm the Freshwater Environment Group Leader at the Scottish Government Marine Directorate. And I also have an honorary professorship at the University of Aberdeen in the Department of Geosciences. So my background was actually on hydrology and interactions between fish and environment. So that's why the thesis is of particular interest to me. But we're also responsible for some long-term monitoring, some trap sites, where we have similar issues in terms of understanding processes and monitoring what's happening in a changing environment.
Thank you. So we also have Eva Thorstad coming from Norway.
Thank you very much. I'm an Atlantic salmon scientist at the Norwegian Institute for Nature Research, and I also work at the University in Tromsø in Northern Norway. Work a lot with fish migrations. I'm very happy to be here. Thank you.
We obviously have the supervisor with an S, supervisors, Etienne Prévost, who's sitting here. You want to introduce yourself?
So I'm Etienne Prévost. I'm one of the two supervisors of Edel's thesis, so together with Mathieu. Mathieu is well occupied with with the connections and the Zoom meeting. So So I will speak later on behalf of the two supervisors. I'm working here at INRAE in Saint-Pétion-Nével, mainly on salmon ecology, demography, and evolution of population facing climate change.
Okay. So what is going to happen now is that we are going to listen to Edel for approximately 45 minutes. And then we will have a short break so that if anybody wants to leave the room before the question, you can do it. And then we will start a discussion with Edel that may take some while. So let me sit here and you can start whenever you want.
So thank you everyone for your presence and welcome. And I want to thank especially my jury for accepting to evaluate my work today. So I am going to present you my thesis about investigating Atlantic salmon migration phenology using a long-term capture-recapture study. So first of all, what is phenology? Phenology is the science of the occurrence of a biological event, for example, the metamorphosis, maturation, reproduction, or the migration, etc. And by extension, this is also the timing of the biological event itself. So let's take an organism, could be a fish, could be a bird, could be a plant, whatever. The organism have a life cycle with biological events. These events are triggered by some cues, which can be external cues or internal cues. So in internal factors, we have the genetics, the epigenetic, the physiological state of the individual, etc. And among the external factors, we have, for example, the photoperiod and the temperature. So these cues have a timing. For example, the photoperiod increases from the winter to the summer and decreases from the summer to the winter. And this timing influences the timing of the biological events. So this triggering mechanism has been selected during the evolution to synchronize the biological event with a more favorable condition for the biological event, which help to reduce the cost of the biological events and to optimize the individual fitness, which can have then impact on the demography of the population. So here is an example to illustrate the importance of the phenology with a well-studied case of match-mismatch theory, the grid-tit on the caterpillar. So the caterpillar hatch during spring, and this is also the timing when the grid-tits breed. And the chick of the grid-tits rely on the caterpillar abundance. They feed on caterpillar. The hatching of the caterpillar is triggered by some cues, and the breeding of the grid-tit is triggered by other cues. In some cases, these cues can make the caterpillar abundance peak and the chick need peak synchronize. This is a match, and this is beneficial for the chick survival and the chick growth. In other cases, the cues make the caterpillar abundance and the chick need desynchronize. This is a mismatch. And this can reduce the chick growth and the chick survival. And as you know, we are in a context of climate change. So to illustrate here is an iconic example of the Japan cherry blossom. So because of its cultural importance, the cherry blossom in Japan has been recorded for centuries. This is the longest monitoring of the phenological events we have. And you can see that the date of the cherry blossom is variable from day to day. But since the Industrial Revolution, there is an advance in this date. This is because of the rise of the temperature and due to the climate change. So you can see that the climate change can disturb the phenology of the species. But to go further in understanding the phenological process and the potential effect of climate change on it, we are going to focus on one biological event, the migration. The migration is a movement of animal between habitats. And there are several species that have migration in their life cycle, mostly for reproductive or trophic benefits. But this life stage is very dangerous. So this life stage is triggered by some cues in the first habitat. And in some cases, these cues can make the specific need of the individual during the migration and the timing of the migration synchronized. This is a match. And sometimes these cues can make the specific need and the migration timing desynchronized. This is a mismatch. So this can have impact on the fitness during the migration or at the arrival. So now, to go further again, we are going to focus on one species, which is the salmonid, the Atlantic salmon. So the Atlantic salmon is an anadromous species, which is an anadromous fish which is in decline for some decades. And here is its simplified life cycle. So the salmon born in the river, the eggs are laid in the gravel in winter, and they develop as fry, and then as juveniles that are called parr. The parr remain in the river from 1 year to several years. And when they are ready, they begin their smoltification. The smoltification consisted in different behavioral and physiological changes that make the parr a smolt that is able to survive in the seawater, in the ocean. So when the smolt is ready, it do the seaworm migration, and when the post-smolt is at sea, it continues its migration toward feeding area. So you can see the migration route here. And for example, here the population of the southern Europe in France and Spain go to the west of the Ireland coast and then to the Scottish coast. They go to the Northern Sea and reach the Greenland. And when they are ready, they come back to their home river in April or in July to reproduce. And the reproduction is in December. So for the following, we are going to focus on this part, the smoltification and the seaworm migration. The migration can be split into several steps. The first step is the preparation. So I told you that the migration is a hazardous life stage. So to be able to survive to the migration and to survive in a new habitat, the individual must prepare before the migration. So for the salmon, this occurs during the parr and the pre-smolt stage, the preceding summer, the summer preceding the migration, and at the end of the winter, just before the migration. Then when the individual is ready, he can go effectively in migration. This is the departure. So for the smolt, this is during spring. Then there is a travel and the arrival. So the arrival is for the smolt when they arrive at sea. And all these steps are triggered by some cues. So especially the preparation of the departure. And according to the literature, the main cues that triggered the preparation of the departure for the salmon are the temperature, the discharge, and the photopellet. And this stage, the arrival at sea for the post-smolt, is critical. This is a very important event in the life of the salmon because there is a lot of mortality when they arrive at sea. And so the timing of the migration is linked with the growth. The more time the smolt the parr spend in the river, the more it can grow. And so the bigger it can be. And the size is linked to the fitness, as you can see in this figure of Simons and collaborators in 2021. So this is the survival probability at sea, and this is the length of the smolts. And you can see that the bigger are the smolts, the better is the survival at sea. And this is why there is a mismatch hypothesis on the salmon. So if the salmon migrate too early, it is smaller. So there are more predators that can eat them. The predation risk is high. And also they can arrive before the food abundance peak. So there could be, this is a mismatch. Then, on the contrary, if they arrive too late, they are bigger, so with lower predation risk, but they can arrive after the abundance peak. And this is also a mismatch. So it seems that there is an optimum timing. So a compromise between too early and too late, where the fish is bigger, so the predation risk is reduced, and it matches with the food abundance. So this is why I am going to focus on the SMART phenology. And more specifically, the aim of my thesis is to understand the phenological mechanism of the Atlantic salmon SMART migration and the potential impact of the climate change on it. But to do this, the first thing is to assess phenology. But how to assess phenology? A very classical approach in animal monitoring, to monitor the phenology of animal is capture-recapture experiment. So in my thesis, I use the data of the capture-recapture experiment in the Scorff River in Brittany. So in the Scorff, there is salmon in 45 kilometers of this river. And I have 25 years of data. So from 1998 to 2023, without 2020. And so here you can see the number of smolt capture per year, which is quite variable. And when the smolt are captured, they are marked. So at the beginning, they were marked with visible implants, then with elastomer. And from 2018 to now, they are exclusively marked with pit tag. And during all these protocols, they were double marked with fin flipping. So this is a heterogeneous data set, which necessitated a lot of planning. And if we focus on the trapping experiment, so we have two traps near the estuary. The first trap upstream is a Les Ménil here that I am going to call the trap 1 in the following. And the second trap downstream is the French Mill that I am going to call the trap 2. So the biological process of interest is the phenology of the migrating smolts. And thanks to this capture-recapture experiments, we have observation of these migrating smolts. However, we have only a partial observation of these smolts. Indeed, if you look at this map, you can see that the smolt can pass through the trap and be captured, or next to the trap and be not captured. So this is, we do not capture all the smolts. Furthermore, the environment have an impact on the biological process with cues that trigger the migration of the smolts. But the environment can also have impact on the observation process. If you look at this two pictures, you can see it. So here it's the trap 2, two pictures of the trap 2 with high discharge and low discharge. And you can imagine that here, this is much more easier for a smolt to escape the capture than when there is a low discharge. So this impact of the environment on both the two processes can causes confusion. This is why the first challenge of my thesis was to disentangle all these processes, the environment effect on the biological process and the environment effect on the observation process. And we've done this in capture experiments based on this part that I am going to explain you now. So you have two traps, first trap and the second trap, and you have smolt that are migrating. Among the smolt that are migrating, some are captured at first trap with a capture probability, and the other are not captured. The fish that are captured are marked, and then they are released. And when they are released, they can be captured at the second trap with also a capture probability, or they are not captured. Among the ones that were not captured at the first trap, some of them are captured at the second trap, and some are never observed, never captured. So we have all these movements of smooths. And in this first part, I am going to use only this part, the capture mark capture experiment. So with this data, we can reconstruct recapture matrices. So here you have the recapture day, and here the marking day. And in each cell, you have the number of smooths that were marked and recaptured. So for example, for the first cell, you have the number of fish that were captured the first day, that were marked the first day, and recaptured the first day. In the second cell, you have the number of fish that were marked the first day and recaptured the second day, etc., etc. And you have this for 1 year, 1998, but also for the 25 years. So we use this data in a mechanistic Bayesian hierarchical model. And so when the fish are captured, they are stressed, and then they are released. And some will continue the migration, but some will take some time before. So to take this into account, we explicitly model this transition probability, this migration probability between the two traps, with a new type of way, a time-to-event process. And we condition the migration probability not only on the marking day here at the first trap, but also at the day of passage at the second trap. And of course, we took into account the discharge effect on the trap efficiency. And what we find is that the discharge have a negative effect on the trap efficiency. And we succeeded in estimating the capture probability at the second trap for each day. So here is an example for 2003 and 2005. So in blue, you have the recapture probability, and the orange curve is a discharge. So you can see that in 2003, the discharge is mostly decreasing, and the capture probability is mostly increasing. This is due to the negative effect of the discharge. In 2005, you have a much more noisy discharge, and so is the capture probability. So, and you can see also that the profile of this capture probability is very different from a year to another, and that there is a lot of variation from day to day. So this very variable capture efficiency causes a very noisy capture data. If you want more information, you can read the second chapter of my thesis or the article that we submitted in the Canadian Journal of Fisheries. So the first challenge was to disentangle the observation process from the biological process. And we've done this for the second trap by using a time-to-event process modeling and by conditioning the migration probability between the two traps, not only on the marking day, but also on the passage day at the second trap. However, we still not have the capture probability for the first trap. So now the second challenge is to model the mechanism of the migration phenology, still accounting for the observation process, including at the first trap. And this is what I am going to explain you in this part. So you remember, we have all these fish movements. And in the first part, I use the capture-recapture data. But now I'm going to use this data to estimate the phenological probability. So we have 4 steps in the migration, but now we are going to aggregate these 2 steps, the departure and the travel. the individual have to prepare before to migrate. So we took this into account with this probability to be ready. So this is the first sub-process. And in the case of the salmon, this refers mostly to growth and smoltification. Then when the individual is ready, it becomes sensitive to the cues that trigger the migration. So for the smolt, according to the literature, these main cues are the discharge and the temperature. So this is our second process. And then the individual do their migration, and we can observe them at their arrival. And for the smolt, this is at the estuary. We capture them at the estuary. But how to model this? So here, this is the migration period. So T0 is the beginning of the migration period. And the migration can be seen as a random event that occurs in this migration period. And to analyze this type of data, what is often used in other disciplines is the time to event process, also called survival analysis in medicine. So here in medicine, this is used to analyze the occurrence of a death or the recovery of a patient after he contracted the disease. So the survival function is the function between the contraction of the disease and the event of interest, the death or the recovery of the patient. And the hazard function is the function that characterizes the probability for the event to occur, knowing that it has not occurred yet. But what we want here is not to use a survival analysis, but to model the migration of the smolt. So we use this type of analysis, this type of modeling to model the migration of the smolt, and more precisely, the preparation process. So the survival function is now the time before being ready, and the events of interest is when the fish is ready to migrate. To do this, we use a weight-bowl distribution, which is a very classical distribution used in this type of model. And the weight-bowl distribution has a hazard monotonic in time. So the hazard depends on the shape, as you can see here. So when the shape is inferior to 1, the hazard decreases with the time. So for a smolt, it would be that the probability for a smolt to be ready decreases during the migration during the migration period. When the shape is equal to 1, the hazard is constant in time. So for the smolt, it will be that the probability for the smolt to be ready is constant during all the migration periods. And when the shape is superior to 1, the hazard increases with time. So for the smolt, it will be that the probability to be ready increases during all the migration periods. So this different shape and different hazard form causes different form of the weight distribution. So you can see in blue here, when the shape is inferior to 1, when it is equal to 1 or superior to 1. And for the smolt preparation, we make the assumption that the hazard is increasing in time, so that the probability for a smolt to be ready increases during the migration period. So this is how we model the first process, the preparation process. And when the individual is ready, this is the second process, the departure process. So to do this, to model this, we use a multiple regression model with a logistic form. And we included 3 covariables: the water temperature, the discharge, and the variation of discharge. And then, of course, there is the observation process, but we have few information on the first trap capture probability. But if you remember, thanks to the first analysis, we have information on the capture probability at the second trap. We have the capture probability for each day at this trap. And thanks to this analysis, we create an informative prior. And so now having information on the capture efficiency of the second trap and knowing the number of fish that were captured at the second trap, this is much more easier to estimate the capture probability at the first trap. And in addition, we also use partially informative prior on the first trap. So here are some results. So here for 2 years, 2004 and 2019. So this is a preparation process. And you can see that due to the way we model this, this process is rather smooth. However, the speed can vary with the year. So in 2019, you can see that the preparation process was much more faster than in 2004. The peak of the preparation process here occurred at the 10th of April, and in 2004, much more later at the 19th of April. Then there is a departure process, and here are the coefficients of the three covariables, the water temperature, the discharge, and the variation of discharge. And you can see that these all three effects have a positive impact on the departure probability. So this is the profile of the migration probability for 2004 and 2019. And you can see that they are very different. In 2004, the departure probability is mostly increasing during the migration period, whereas in 2019, there is some peak here. So there is very different departure probability profile. And thanks to these two submodels, we can we can reconstruct the phenological probability that you can see here. So in 2004, we have a unique peak quite late, whereas in 2019, we have several peaks, with the first one much more earlier than in 2004. So our model succeeded in recreating these different migration groups. And of course, there is still the observation process upon this. So we succeeded in estimating the capture probability at the first trap and at the second trap. And so here are the data, the raw data we had at the first trap. So this is the frequency of capture at the first trap in 2004. And here, this is the frequency of capture at the second trap. So you can see that they are a bit different. And here is the phenology that we estimated. And you can see that it seems that the phenology is quite similar to the frequency of capture, a bit of compromise between the two traps. But in fact, if you look at the QQ plot here, the phenology is quite different from the capture. So here, you have the cumulative capture frequency for the first trap and the cumulative distribution function for the phenological probability. So if the raw data and the estimation of the phenology were similar, the circle here would be on the line here. And you can see that it is not the case. The phenology is much more faster than the raw data at the first trap. Here is a second example for 2019. So still the frequency of capture at the first trap and here at the second trap. You can see that the data are very different between the two traps. There is an intense peak at the beginning and at the end of the migration period for the second trap, and not for the first trap. And so the phenology is a bit different is very different from the frequency of capture at the second trap, and still a bit compromised between the two. And if we look at the QQ plot, we can see this difference here, which is less at the end of the period. And The third example is in 2001. I show this example because you can see here that the frequency of capture at the beginning of the period is equal to 0. This is because the trap was not functioning at the beginning of the migration period. So we do not have capture for the first trap at the beginning of the migration period. And thanks to our hierarchical approach and the information we have at the second trap, we succeeded in reconstructing the phenology, even if we did not have data at the first trap. So to conclude on this model, we have the raw data that are very noisy. And this is because of the observation process, which is very variable from year to year and day to days. And this is also due to the biological process, which is also noisy. And this biological process is due to two sub-processes, the preparation and the departure. So by explicitly reconstructing all these multiple layers, so the data are a result of multiple layers that we succeeded in explicitly reconstructing. So the second challenge was to model the mechanism of migration phenology, still accounting for the observation process. So we did estimate the capture probability for the two traps this time. And we also explicitly model the mechanism of the migration process and succeed in explaining this migration pulse of GF27. And now, what about climate change? So here you have the migration window for each year. So this is the 25% quantile of the phenological distribution to the 75% quantile. And the circles are the median. So you can see that these migration windows are very different from one year to another. Sometimes they are longer, sometimes they are shorter. But there is no specific trend, like they are not increasing or decreasing. However, if you look at only at the 25% quantile of the phenological probability, you can see that this, for the last few years, the phenology is getting earlier. The beginning of the phenology is getting earlier, which is consistent with the literature. For example, with the meta-analysis of Otero and collaborator in 2014. So in this meta-analysis, they aggregated the raw data of many capture experiments in many countries, in Europe and in Northern America. And they show that this migration is the 25% quantity of the capture data is getting earlier. But how can we explain this advance? So first, to better understand the role of the preparation process, I ran different linear models with different covariables between the quantile of the preparation process, the quantile 25%, and so different variable. And the best is this one. So this is the mean water temperature in March, so just before the migration period, And you can see that the warmer is the water, the faster is the preparation process. And we can assume that this is because of better growth condition. If the water is warmer, the smolts can grow better, and so they may prepare faster. Now, for the role also of the departure condition, I also run linear model with different covariables between the 25% of the phenology distribution. And so the best is this one with the mean water temperature. from the beginning of March to mid-April. And you can see the same, that the warmer is the water, the earlier is the phenology of the smolt. So I have this model, and I have also the projection of the water temperature in the scope until 2017, thanks to a hydrological model of Guillaume Tirel. And so I used the coefficient of this projection and the water temperature to make projection on the smolt migration phenology. Here are the results. So here we have less intense scenario of climate change. So you can see that the 25% council in the model, estimated by the model, the mean is on the 13th of April. And here with the projection, the mean between 2075 and 2070 is on the 10th of April. So this is 3 days earlier with this scenario of climate change. This is worse with the middle scenario and even worse with the worst scenario of climate change. So we, instead of the 13th of April, this is the 7th of April, so almost a week earlier. And could this have impact on the fitness? So there is a change in the phenology smolt migration, but there are also change in the ocean. And if the change in the river and in the ocean does not correspond, there can be a mismatch. But I have no data to test it. However, there are studies that are recent studies, for example, here in 2024, that are studying the impact of the arrival at sea of the salmon and the availability of food. And you can see that the survival of the salmon is better when there are more food available. So yes, the mismatch is a risk that can influence the fitness of the smolt. The other hypothesis is that if the smolt migrate too early, it can be smaller. This is not what I found in my data. I found that the earlier years are associated with better factor conditions. And with our results, we can assume that this is because we have, this year, we have better growth conditions. So the smolts can grow better on our year. And I also ran linear model between the return rate, so as an indicator of survival, and the 25% conceal of the phenology, and I did not find any significant effect. So with the results we have and the data we have for now, we cannot find any impact of the phenology advance on the fitness. So in my thesis, I explicitly modeled the observation process and the biological process to disentangle all these effects. I also propose a mechanistic model that is flexible enough to reconstruct the different pulses of migration of salmon. And I believe that this model can be extended to other populations of salmon, but also to other anatomous fish and maybe other species, maybe, for example, birds, migrating birds. And I also found that the salmon migration is getting earlier and earlier. And this trend may continue in the future. But to better know this, we can improve the model we have. For example, by better having a better explicitation of the preparation process. So I show you that we modeled the preparation process with distribution of Weibull, a quite simple distribution. And but we can also integrate variables into this process, such as, for example, the water temperature or the charge during winter, etc. And this would help to better understand the link of between the preparation process and the climate change. Having this better model, we can do projection. So I show you projection with a very simple linear model that does not explain a lot of the variation. But we can use our model with also this advanced to do projection with the projection of water temperature and discharge we have, to do better projection. And if we have more data about the ocean conditions, such as the food available, the temperature at the sea entry of the smoke, etc., we can study if there is a potential mismatch. And to finish, we have also individual data that I did not use in my model. So we can extend, after this, we can extend the model by including other processes and including this data, for example, the evolutionary processes, etc. So now I just want to take some time to thank everyone that helped me in my thesis. So first, I jury member for being here or online today. I also want to thank Mathieu and Etienne. My thesis would not have been possible without you, so thank you. Of course, I want also to thank the SCARF team, and especially Nicolas Janot, Fabien Kendo, and Johan Guillou, for your welcome, your help, and for collecting my data. I also want to thank the member of my committee, Etienne Rivaux, Cyril Piou, and Colin Bouchard, for your advice during my thesis. And because in my thesis, I participated in a lot of things, many adventures, a lot of training and organizing of seminar and workshop, etc. I want to thank all the PhD students that helped me in doing this. Thank you for everything. And of course, thank you to all the lab for the great environment, for your conversation, for your support, etc., etc. And of course, thank you for your attention. And I'm now available for questions.
Thank you, Edel. Perfect timing. So we have a very short break for those who want to leave the room and for me to get my computer. Okay, should we resume? Okay. So since I'm also a reviewer of your thesis, I'm going to start. Let me apply my own rules to myself. Okay. So first, I'm going to start with congratulations for this presentation. And also, I wanted to thank everybody for inviting me to be on this committee, because I really enjoyed reading your thesis, and I found it very impressive. And one of the reasons is that I'm not a salmon person, so I didn't know much about salmons, Atlantic salmons, before reading the thesis. And I'm not a statistician either. So when I got to the final chapter and when I realized that I had understood something, several things, hopefully, about the topic of your thesis, I realized that you have done a very good job at explaining complicated things. And also, concerning your presentation, it helped me a lot understand transitions in your manuscript that I had not understood before. So this is a quality that we can talk about later. I'm going to start with some general comments because I wrote a report that not everybody has read here. So first, if I'm one of the reviewers here and I've recommended that you defend your PhD, this means that I believe that there's a good chance that you get your diploma because of the quality of your work. So this manuscript meets the standards of a French PhD with a very high, with a paper in very good shape submitted to a high standard scientific journal. And another one that is also in good shape, I find, but perhaps a little bit too long. I don't know. The different chapters are based on the analysis of a very long-term data set, a very impressive data set. So I understand that this is a teamwork, but you have been analyzing the data and have developed complex models, mark recapture models, and you have estimated the parameters of those models using in the Bayesian framework. Apparently, if I understood correctly, your models in the first model you developed relaxes assumptions that were made by previous authors and that you found not desirable. And concerning the other one, the second model, the phenology model, I don't know if there is anything similar in the literature, but apparently this is very, very novel. Another quality of your manuscript is the introduction, a very thorough synthesis of what phenology is. And indeed, you have not talked about salmons exclusively. You have talked about birds and tits. And so this is, I think this is a good thing to do. And you have also worked on the definition of the word of phenology, because this may change according to the study system. Yes, I appreciated this background in the introduction and the background concerning the Atlantic salmon, because this is a very fascinating life history, but if you don't know it, you don't understand anything in a thesis like this. So I'm going to try to ask a couple of questions. Even though this is really not my area of expertise, and my questions are probably not very consistent with one another, but anyway. One thing that I wondered when I read the chapter 3, the phenology chapter, right, is the reason why you decided to separate the two processes. And so you have one process that is the preparation process, which is not observable. And you have the other process, which is the start. The bird starting to move, or starting to the journey to the sea. So I understood that some other authors have used the same approach, like Pulkkinen. Kind of. So could you explain to me why you decided to do this, and also, well, okay, if this author has not proposed the same approach, there must be something different with previous work.
So there are yet several studies on the phenology of Atlantic salmon, and but mostly they are only looking at the raw data and not trying to explain the mechanism of the migration. So to do something new, I wanted to really model the mechanism to better understand the process, the biological process. And so this has been made by a few authors, such as Pulkkinen, but not exactly in the same way. And so I choose to separate the preparation and the departure, because it is two of the main steps of the migration. This is a very general model. So it also a general step. So this also helps the model to be very generic. It can be also applied to other species. So this is what. So the first reason is to propose something new and better understand the phenology of the salmon. And the second thing is to make the model general and applicable to other species. Yeah. I don't know if you're...
Okay. I understand why you would like to separate the preparation step and the actual journey. Because including in birds, I mean, some birds have to store fat before, before deciding to move. So what you are saying is that this decision does not depend on the specific setting that you have, the experimental setting that you have with 2 stations, and so this has nothing to do. It's general enough to be
for the biological parts. No, I think it's general enough to be applicable to other anadromous fish, and maybe also to just the biological part to bird, for example.
Okay. Well, if this is true, this is going to be very interesting in the future to see the future applications, including songbirds, stopover, migration, and so on. Okay, thanks. Now, obviously my questions are completely disconnected from one another, but another question that I had concerning this paper is the way you used the phenology, the estimated phenology in the discussion to test hypotheses about the influence of temperature, water temperature, and discharge, I think, and maybe one other photoperiod. No, No.
No, the variation of discharge.
Right,
The
the
variance in
variance,
the-
yes. So you are using, so first I would like to understand, make sure I understood what has been done. So you have estimated the parameters and also the quantiles,
Yeah, so the,
and then you have built models using the mass package, using those parameters.
so I took the phenological probability and I, so the posterior of
Right.
this phenological probability. I took the median of the posterior and I took some variable of interest and so for example the quantile 25%
Right.
of the probability of phenology. And so I use this as a true phenology of the smolt, as if this is the phenology of the smolt. And I want to explain how the causes of the variation of the phenology each year. It's
Okay.
more clear on it.
Yes, yes and no. Because, okay, so my question is why were the discharge variable and the temperature variable not included in the model for the phenology?
Yeah, they are included in the departure process, but not in the preparation process.
Okay.
And why? This is because I had only 3 years, and
So it's a matter of time
yeah.
limit.
So this is why this is a perspective to include covariable into the preparation process, but it's very challenging. So we did not have the time to make this, but it can be done in the future, or at least we can try to do it in the future to better explain the preparation process with the temperature, discharge, etc. And
Yeah, okay.
also, there were many questions about which, for example, which temperature to choose and to put in the preparation process. For example, we know that some studies use the degree days for the smoltification, so we could use the degree days, but since when? So there were many questions that must be answered before to be able to model this. So yeah, we did not have the time, but that's the next thing.
But well, thank you for your explanations, because I started wondering if this was a technical problem, if it was too complicated. If you're saying that it's just a matter of time, okay, this is.
At least we can try.
This is understandable. I mean, you have only 3 years for your PhD in France, so and perhaps I expressed my question in a bad manner, but indeed, what you are interested in is the preparation process. So you would put covariates on this unobserved process.
Yeah.
Okay, this is even more interesting.
Yes.
Okay. So this brings me to another. It's not even a question, but perhaps this is something that we should discuss after the defense. But when I arrived here this morning, I asked to Mathieu if he had written a paper a long time ago using an approach based on a latent trait, the latent threshold trait. So this is basically an observable trait. And this approach is currently being used to model migration phenology and breeding phenology in birds. So it's something that you cannot observe. It's some sort of intrinsic, unobservable individual trait.
Yeah.
And that can be directly influenced by either intrinsic or extrinsic factors. So Mathieu said, yes, I did that when I was younger. But when I started reading your thesis, I said, I saw that you would model something that is not directly observed, that you called a latent process. And I thought, oh, they are using this one approach used with birds. So do you think this approach could be interesting in your setting?
In my model?
Yeah, I mean, to study migration.
Yeah. So I don't think I have read this, but I think
it's a latent trait approach.
Yeah.
So
this is one of the strengths of this hierarchical model, to be able to reconstruct or try to reconstruct all this latent layer. And I think this is very interesting because in biology it is difficult to observe everything. So yeah, I think basing our approach on knowledge we have on the mechanism and reconstructing these layers, I think would be very interesting. But
yeah, okay. Well, perhaps we can talk about this on Mathieu after the defense. Just that I was wondering again if it was a decision not to use this approach that is currently being developed for migration phenology in birds in Norway, in fact. The group that is working on this is in Trondheim. So we can talk about this later. Okay, perhaps one more question on okay. Yeah, in fact, two questions. In the discussion, you explained that the migration pattern in the Scorff River follows patterns that have been observed by others.
Yeah.
Earlier migration, especially in the earliest stages of migration. And that there is a strong relationship with water temperature and also discharge, but maybe not as strong as we thought. And then you say in the second part, for the second part of the migration process, there may be more variables involved. Like, okay, the second part of the migration process is less variable, which may be due to the fact that this part depends on a greater number of variables. This is in the thesis, and I was wondering what.
I was speaking about the second processes, the
Yes,
departure.
probably that's why I understand. That's page 188. So a greater number, is it 88? Yeah. I was wondering which variables. 88. Oh, it's in the conclusion. Yeah. Maybe the second part of the migration period. Oh, sorry.
Yes.
Period.
Okay. Yeah. The second part of the migration period. So I was referring to the other contents. So it's really the 75% and the 19% content. So the end of the migration.
The late, late ones.
Late smolt. And I think this latest smolts may be affected by other variables, which are more biological. For example, their age. We know that often the 2-year smolts migrate earlier in the migration period than the 1-year smolts. And we did not take this into account in our model. We aggregate all the age. Maybe also the genetics. There are studies. There is no studies yet on the Atlantic Salmon. I think there are one that are currently run on the genetic basis of the smolt phenology. But we know that the phenology of the migration in other salmonids is partly genetic. So there may be smolts that are earlier smolts or late smolts due to their genetic, but we did not took this at a point. And also maybe their ability to grow, because the smolt do compensatory growth just before the migration. So if they're able to do it or not, if they were maybe too small at the end of the summer, so they have more growth to do before migration. So yeah, I was thinking of more biological, like internal factors that we did not take into account in the current model.
Okay, thanks. My last question is somewhere here. Yeah, in fact, you mentioned this in your presentation that early migration doesn't seem to be associated with poor body condition. Okay.
Yeah.
So, which apparently came at a surprise compared to other results obtained by other groups.
Yeah.
So, if they are in good shape, in your opinion, what would be the most urgent thing to study now to project the possible impact of climate change on the future of this population? Okay, you have a lot of money to invest into a project. What would be your priority?
Okay, so I think we don't know enough on the C part of the life cycle. And so if it was possible, I wish we can tag the smolt with tag storage, data storage tag. They are quite heavy, so for a smolt it's complicated. But if we had we could, we can after when we have the tag back, we can make model to know where they are going exactly, and study the potential mismatch with the data, because there are data on the zooplankton on the stock of fish that smolt feed on. So maybe with this data and the exact or almost exact migration routes of the smolts, we can study if there is a potential impact of on the smolt fitness. And yeah, but yeah, this is technically and economically complicated.
Well, okay. Well, thank you for your answers, and again, congratulations on your work. I'll stop here and ask to Samu if you are ready, if you can.
Yes, I'm ready here. I suppose. Am I on your screen or not? Can you see me?
Yes, you are.
Okay, nice. May I ask if Edel could move a bit closer to the microphone, wherever it is? Because it seemed that I was hearing the questions better than the answers. I could hear everything, but there was a slight difference. Okay, then. Yes, I would also very much like to thank you for inviting me to do this reviewing and being your opponent here. And of course, when I started to read the thesis, I was kind of pretty quickly noticed that I understand why did you want to ask me here, because it seems that you are building on much of the work that I did about 2 decades ago when I was doing my PhD at the time and developing the mark recapture models for salmon migration. And now, after reading the thesis, I can really say that this is there are big steps forward which you have taken, and I'm very glad to see that things will progress. And I think you have done I think the word previously was impressive, and I think that's the right word here to kind of describe the amount of progress and novelty that there is. So I'm very pleased with the work you have done. Of course, in this situation, we also need to discuss a bit about the potential critical points as well, which I was also able to find. So we have at least something to discuss here. And maybe we could start with the kind of philosophical questions first. And this is something you didn't mention too much in your thesis and not in your presentation, that why did you choose to use Bayesian inference in the first place here? What's the point? Why?
Okay, I think for this type of analysis, Bayesian is nice. First of all, because a lot of capture-recapture study are run into Bayesian. So I think there are quite a lot of literature on it. So this is practically a good thing. Easier to base your thinking your reflection on the literature. And also, I think the Bayesian is allow to expand the data, like to the information to. There is uncertainty. The uncertainty is much more easier to take into account in Bayesian. The fact that you can treat prior with preceding analysis. You can use this as information in more informative prior as other information in your model to add information. So this, I think the Bayesian is quite well adapted for this type of ecological data that are very noisy and with a lot of uncertainty.
Okay, thanks. Regarding these uncertainties, one of the things that we notice in your analysis here is that you of course have to use the concept of probability in 2 different kind of ways. And one is, for example, these capture probabilities, migration probabilities, probability to move. But then on the other hand, you put probability distributions on those probabilities. So you have a situation where you have a probability on probability. Can you somehow explain that situation, what we should understand here when we have a situation that we have probabilities on probabilities?
I'm not sure I see where what you are referring to.
Let's say, for example, that I have a capture probability of smolts in some day, and then I put a beta distribution as a prior for that capture probability. And this is a probability distribution that I put on this another probability. So can you somehow explain that what's the difference of the probability that we put on the capture probability or the capture probability itself? Or do you see any difference there? Kind of philosophically, what do they represent?
You mean the... Oh, okay. So you mean that to model the capture probability, I use probability, that's it?
Yes, you put a prior distribution, like a beta prior distribution on that entity.
Yeah. So what's the difference between these two types? The prior is something we choose, and that can impact the analysis. And the capture probability is the process, the real process we want to estimate. So these are two different things. The prior is something we choose, and the estimation comes from the data.
Yeah.
So, but in the analysis, they are dependent, like the yeah, the prior is much more theoric than the estimation probability, like the capture probability. I'm not sure I have answered your question, but
yeah, I think you are getting there. Yeah, it's good. So if I understood you correctly, you are saying that now the capture probability is something that we think that describes the actual process in nature.
yeah.
And then, but can you somehow elaborate what do you actually do when you choose the prior? What is the prior now for this model parameter, we could say? Where does it come from? The choice, what does it mean?
Yeah. So the posterior of the capture probability will partly depend on the choose of the prior chosen, and depending on the quantity of data that there is in the quantity of information that there is in the data. So I lost my way. So yeah, the prior could have an impact on the posterior, and this is why we must choose it with caution. And for example, we have to choose a prior that correspond to the value. Like for example, the probability is between 0 and 1, and we could not, like if we put a prior very large, this is nonsense. Like a prior between negative value and big values. So we have to make this prior logical with the parameter we are estimating. And also if we have information on or if we have prior information on the parameter we want to estimate, we can put this information in the prior. But we have to check that it is not, there is no conflict between the prior we choose and the information that there is in the data.
Okay, would you agree if I would say that the prior distribution then describes what you know currently about this model parameter? So the probability of capture is a model parameter. We think that it kind of exists objectively. We could say that there is such a thing, but then when we put the prior on that, we are effectively using now a concept of probability to say what do we know about this parameter. Would you agree with that?
Yes.
Okay, good, excellent. Then we agree. It's nice to agree. Okay, then onwards with this kind of different definitions. Now we have been discussing what's the Bayesian inference, because you said that you are doing Bayesian inference. Now, what's the difference between mechanistic modeling and something other, like statistical modeling? Why is it important? What is it? What's the difference?
Yeah, mechanistic model is a model that try to reproduce the mechanism, that try to explain the mechanism. For me, a mechanistic approach is much more general than a statistical approach. A statistical approach can be applied on one set of data, and if we try it on another set, it may not function. So we choose another two, and so it helps to know to learn about the data sets. But this is not very general. This may not be sufficient enough to understand what is going on, because there may be confusing effects and stuff that we were not aware with this statistic model. So the mechanistic approach is a way to model, to better reproduce what is going on effectively in the process. But I think this is quite hard. This is a bit hard to do. So I think this is why mostly we use statistic approach, but sometimes mechanistic approach can help to be more general and better understand, for example, make better projection of the process of interest.
Is there a difference that where does the information come from? How do you formulate the mechanistic model compared to statistical model? Which are what's the kind of basic idea? How do you come up with a mechanistic model compared to a statistical model? How do you do that?
You mean in general or in my thesis?
You can answer whatever you like, whether in your thesis or generally. Yeah.
Okay. I will take the example of my thesis. So as I said, there is yet several studies on the phenology of Atlantic salmon, but they took a simple statistical approach, and sometimes there can be you can have confusion due to the factors that are involved in the process. I have the example for the grid teeth. Most models take the temperature for the breeding of the grid teeth phenology. Most models take as a cues the temperature in a specific period, but they say in the papers that it may be not the cue, but a cue that correlates with the real cue. But doing this, and for example, making projection, we could not assure that the relation between the real cue and the cue we use in the statistic approach will continue in the future. So this is why a mechanistic approach, basing the approach on for example, the physiology of the grid teeth. If we know that this cue have this impact and this trigger the breeding of the grid teeth, we can much more easily predict the grid teeth phenology in the future than with a statistic approach that may contain some confounding effect.
Yeah, thanks. Would you think that it would be a summary of your thoughts that if I would say that the statistical modeling starts from data and tries to kind of find patterns in the data that is supplied. And then the mechanistic modeling is that you use your biological knowledge and expertise and biological theory to formulate the causal dynamics of the system without actually seeing any of the data even.
Yeah, I think this is quite this. And this is why also we cannot make a mechanistic approach in every case. In the case of the salmon, this is great because we have a lot of study on the salmon, on the life cycle, so we can propose this generic approach. But there are some species, some place where we have very few information, so a statistic approach is the best to get knowledge before to really seek into this process.
Yeah, good. Then a question about the concept of parsimony.
Yeah.
How would you describe what it is, and is it different when you do mechanistic modeling and statistical modeling? This is something that you quite often refer to in your chapters, that you do a certain kind of modeling choice because of parsimony. So what do you mean by that?
Okay, so the concept of parsimony is to not use too much parameters for the to include not too much parameters in the model, because it would be hard to distinguish these parameters, to not have confusion between the parameters, and this is not. We can have over like over fitting. But so yeah, we have to make choice and not take as many parameters as we would like. But I think this is a bit different in mechanistic approach. I think we can add a bit more parameters in mechanistic approach than in statistic approach, because we base these parameters on biological information we have yet. And we want to explain this mechanism and not just to get information, to get knowledge, because we know nothing about this mechanism. But still, we have to check that we can effectively estimate these parameters and know that there is no big confusion between two or several parameters, for example.
Yeah. Well, what bad would happen if we notice that there is no update in some of the prior distribution, or there is confusion between parameters? What is the kind of bad thing? Why we would want to somehow evade such a situation, even in mechanistic modeling?
Because in this case, we could not know if the parameters is relevant and if the process we are modeling is true, if it's relevant. So we want to make the estimation of the parameters more the more precise, without too much confusion.
What if we have a situation where we think based on the biology that we need these parameters and they are relevant? Then we find out when we look at our data that now we actually can't estimate them very well. And if I now then say that, okay, now I need to simplify the model which describes reality, what then happens to my estimation of uncertainty when I do this kind of thing, that I make simpler model because I couldn't estimate the parameters of the model that I think is realistic?
Okay, so I think it will be a choice of the person who is doing the model. Like if the person think that their model is really the truth and that there is just not enough information in the data, so we should get more data to confirm the model, for example. Or if the person prefer to say, okay, I'm not sure of my model, I thought it was the true, but with the data, with the lack of updating I have, I'm not sure anymore this is the true model, and I want to simplify the model to make sure. So I think this is really a choice, and this is context dependent. If we have, for example, in the case of the salmon, we know a lot. In the case of another species, if we know very few, and maybe it would be better to simplify the model.
Yeah, thank you. I think this is a good answer to this question, that it's context dependent. Sometimes you kind of have to do things which you wouldn't do in another situation. Thank you. And I think my time is probably up now, and it's time to continue. I once again want to congratulate you on this excellent job, even though we went into kind of hard questions here. I think overall this was excellent job that what you have done in your thesis. So congratulations, and let's give floor to the next one.
Thank you.
Well, thanks a lot for your questions, Samu. And so we decided that the next person to speak is Ian. So please go ahead.
Thanks very much for the invite. Yeah, and thanks also very much for sharing your thesis with me. I found it to be very well written, really nice introduction, really broad, nice introduction. Incredibly challenging, thought-provoking modeling that really got me scratching my head a lot. And the whole way through, I kept asking questions all the time, and then you answered a lot of them in the discussion. So that was good, you know, because a lot of what I was constantly thinking about was answered really nicely at the end. I mean, I think from my perspective, working in similar problems, similar rivers, there were a lot of challenges there, and you had me thinking a lot about my own data and what might be the challenges around that, and in particular, being able to sort of separate the observational process from the underlying process of interest was hugely interesting, and to illustrate that at the end, I think was really great. So yeah, you know, overall, I thought the thesis was really excellent, and like one of the much better theses that I've had the sort of joy of reading. I also really liked the way that you illustrated the statistical models. So sometimes I might find it a bit challenging to understand exactly what the models were, but I think that your illustration of the model outputs was really great, you know, and that really helped me a lot. So thanks for that. I think I might like to sort of try and just ask a few questions, sort of stepping through the process, maybe starting with the capture probability sort of element of things. And probably the questions maybe a little, because of where I'm coming from, they're maybe more, some of them are maybe more a bit environmental, and probably a little bit less philosophical than some of the other ones, maybe more specific. One of the things that got me really interested when I was having a look through some of the modeling was when you were estimating the capture probabilities and the average ones. Maybe you can share it with you. So great. So I was really interested that there appears to be something in the early part of the period, like 1988 to 2010 or 2011, and then like it looks like the capture probability is much higher, and it looks like it's much more variable, and then it looks like it sort of stabilizes, and this sort of struck me in the data, and I'd be really interested can you tell me anything about what might have happened there, and also could you tell me whether that has any implications for them when you estimate the other effects, like the ones of discharge? Is there any not cross-contamination is not the word I'm looking for, but like in these years, do the discharge effects behave in a different way from in the years where the capture probability is more generally lower?
Yes. So I may re-ask you the second part of the question.
Yeah.
For the first, yeah, we were a bit surprised of this part of the higher probability, capture probability. But there is several things we don't know about the trap functioning. So this was the beginning of the protocol. So they were still they were trying some to change some parameters, etc. So maybe the trap was more attractive with the parameters, with the way that the trap was at this period. Also, there is, for example, there is vegetation and sand just before the trap, and it change every year, and we do not have data on the form of the sand and the sandbank and the vegetation. So maybe it can also increase or diminish the capture the trap attractivity. The trap, yeah. So we don't know exactly why there is this higher capture probability at the beginning of the protocols, but it may be due to all these parameters. Also, there is a weir before the trap, which is private. And so we don't have any data on the way that the people was using this weir. So maybe also it can have consequences on the hydrology, the hydrodynamics, and make the trap more or less attractive. So we assume that this is because all of these variations that we do not have any data on, and we could not explain that this is because of all these parameters. And so the second part was
So I guess I was just wondering whether there was any sort of risk that, for example, when the capture probability is high, then there's some knock-on consequence for your estimates of the discharge effect, and then if it's low in the opposite direction, is there any?
Yeah. So the discharge effect is so there is a different discharge effect for each year. So it's allow the model to accommodate for the effect of the higher capture probability and the effect of the discharge. So I think it can buffer the effect. And after, we haven't checked further if there were particular effects of this intercept on the discharge effect. We haven't checked this like intentionally. This is what you was referring to?
Yeah.
We haven't checked, yes. But yeah, that's interesting.
And then I was wondering, some years you have a small positive effect of discharge on capture probability, and most of the years a negative effect.
Yeah.
And I was wondering, is there any mechanistic reason that you can think of about why in some years you might have a small positive effect of discharge on capture probability?
So I thought that maybe these years were a year where there were very low discharge, and if the discharge was a bit higher, the trap attractivity was better. So I tried to make this with a square effect of the discharge, but we did not find any significant effect, like any, it was not conclusive, so we thought maybe not. But so I cannot explain it with certainty. But I think this is also due to parameters that we have not took into account. And yeah, I think sometimes this is if there are stuff that were moving around the trap, and when the discharge was low, the trap was not attractive at all. The smoke can easily go back and pass through the other way. Whereas if there is yeah, so it could be an interaction between the trap attractivity and the smooth behavior and capability to swim and not being trapped into the trap. Maybe. I don't know.
Yeah, no, it's fair. There's lots of reasonable.
Yeah.
Yeah, lots of reasonable. Yeah.
Maybe so with this analysis, an antenna, acoustic antenna, have been installed, set up near to the trap. And maybe with this, we can also know the behavior of the smoke before the trap. So maybe we would have some answer in the future, maybe.
Yeah, so that might be some way of improving things, improving your understanding, or
our understanding of this effect, of the trap attractivity and the small behavior when they are marked or not. Maybe.
And the discharge data that you feed into the capture probability model, and then also feed into the smooth migration model.
Yeah.
Can I just ask, like, where is that collected relative to this location?
I was sure you question me on this. So the discharge is the data are collected just in another mill before the two traps. So this is quite close to the trap. And there is no other river tributaries between the place where the discharge is collected and the trap. So yeah. This is not exactly at the trap itself.
So maybe this is an unfair question, but do you think the discharge data are more representative of the emigration process or representative of the hydraulics at the? So I totally understand you used the data that you had available to you, but is there a chance that effectively the discharge data are don't well represent the conditions actually at the trap? Therefore, that's why you get such variable parameter estimates on the capture efficiency.
Yeah, I think indeed the discharge is more representative of the migration probability because it's a bit upstream, and so are most of the smolts that are migrating. So this is more representative of the condition in the river and not at the trap specifically. But this, we hope that this is quite representative too. And still, this is an approximation of the discharge. And I hope that the discharge at the trap and the discharge that we are collecting is quite consistent with each other. I hope, but I cannot give it.
So that I was also wondering with you having sometimes a positive parameter estimate and sometimes a negative, but most often a negative.
Yeah.
Did you also try some, like, or are you able to incorporate nonlinear responses into your models, or did you explore that at all? Like a modal, say some sort of reverse modal response, or?
Yeah, nonlinear model on the capture probability we tried.
Right.
But we haven't tried with the current version. But we tried before and it was not consistent, but we thought that it could be, so I don't know. I have also tried to see if this year where the discharge effect is positive is the year with more extreme discharge or less extreme discharge, and I haven't found anything like it would have needed more investigation, and I haven't I had no time to investigate it more. So yeah, there are stuff I cannot explain in this capture process.
So if you wanted to improve this bit of the model,
yeah,
are there any ways you could think of to collect more data that might help you better understand what's happening?
We could collect the discharge just at the trap and see if there is difference or not with the discharge we have currently. And I don't know if maybe the people that work on the smart ability to swim and stuff can also do something with to see if there is difference for the attractivity of the trap. But this is not my my field, so I don't know.
Yeah.
Yeah.
No, that's fair. In terms of where you stepped on from the capture probability and the larger models on the emigration, I kind of wonder, like, could you step me through the thought process that led you to create the models in that way?
Sorry, can you repeat?
Yeah. So when you in the next chapter, when you start to think, I want to represent this process and this process and this process, could you just, like, in general, just step me through what's the thought process? What led you to think that what you did was a good way to model, this process?
You mean by separating the preparation, departure, and observation process?
Yeah, I mean, it's obviously very straightforward because in your thesis it all works well, no issues and no problems.
Yeah.
So I presume that's the way it started, that it just was a straightforward, you got it all right straight away, or can you say something about kind of what was involved, how you thought about putting it together and how things might have evolved a bit?
The refers also to the fact that we begin with the observation and after with the biological process.
Yeah, so I guess I'm thinking, at the outset, somebody would have said to you, right, this is the problem we're looking at. These are the data, and then you have to think, okay, how do I move on from here? How do I, what sort of structures do I want to live with? So I'm just keen to get some insights into how you thought about approaching this.
So when I began my thesis, Mathieu and Etienne and the people that work there really think that the discharge has an impact on the capture probability, but it haven't been really modeled. So this was, and thanks to the double marking, we could estimate this capture probability and the discharge effect. So this is why knowing that the discharge have an effect on the phenology and suspecting that the discharge has an effect also on the capture process, we begin with this experiment to first model the capture probability and then the phenological process. In fact, it took much more time than we hope, and so we spent 2 years developing the capture mark recapture and even more model. So this is why we the first thing was to know better the capture probability, because we wanted this, not the total estimation of the smolts that were migrating during the period, but the quantity of smolts that were migrating each day. Because to study the phenology, we wanted very discrete. So we wanted to have the capture probability per day to reduce this confusion. So this is why we begin with the capture mark recapture model. And after, we include the biological model. And so we choose to separate the processes, as I explained to Emmanuel, to be more general and be more mechanistic approach and better explain this mechanism.
So and that was a good discussion earlier. I think multiple times about the readiness sort of side of things. And so I guess my next question that feeds on quite well the next questions, which are around some of the temperature responses to migration. And one of the things that I've always thought is that there's a huge problem in separating out like the day of the year effect, like something, it's just time. It's just like the fish eventually are gonna go out, you know?
Yeah.
And the true effect of temperature, when the two things are like so strongly correlated, you know?
Yeah.
And in all of the analyses I've seen, that's been like a big challenge. So I guess my question is around the separation of time from temperature effects when the two things are correlated.
Yeah.
And how confident are you that by separating out the, you know, effectively this temporal process and then the temperature process, that you're really capturing the effect of temperature, because it's such a big effect in your models on the migration time. Are you really confident it's the big effect? It's like much bigger than the discharge effect and so on, but also that you've managed to separate it out from this time effect.
So we did not explicitly include the time in the model in the way we model this because of this correlation, and we could not it was not very distinctible to estimate to separate the effect of temperature and the effect of timing. Because yes, there is correlation. I check it, so there is more or less correlation depending on the year. But there is correlation, of course. The temperature is mostly rising during this period, as this is spring. So I think the effect of the temperature is not the effect we find is not exactly. It's not perfectly only the effect of the temperature, but there might be a bit of confusion between the effect of temperature and the effect of the photoperiod, for example. And this is why we haven't put the photoperiod explicitly also in the model, because this was correlating with the temperature. And so there might be a confusion, but we also know that the temperature has an impact on the smoltification of the smolt, because of other studies that study the experimentally the effect of temperature and the effect of photoperiod and their interaction. So we know that the temperature has an effect on the departure, and we also know that the warmer water temperature, to a certain extent, is good for the fish for their swimming ability. So yes, there might be confusion between the effect of temperature and photoperiod, but also I think there is really an effect of temperature, because we know that this amplified the smoltification and the smolt swimming abilities.
So the temperature effect in your models, do you think that, and the temperature effect on like probability to migrate out, do you think that the temperature effect is describing both something about the trend and something about the day-to-day variability, or do you think it's really capturing that day-to-day variability?
I think it also captures the day-to-day probability, because I want to go back to the 2 years I have, 2004 and 2019. So you can see here, this is quite, I think this is mostly because of temperature that is increasing. So you have a departure probability that is mostly increasing. But in 2019, the temperature was not that much increasing, and we have Oh, yeah, it was increasing, but yeah, we have different. No, sorry. The temperature was still increasing, but we have different peak here. So yeah, I think there is also, this capture also a bit of the variation from days to days. And I think also when I look at the data, for example, in the capture data, at the beginning, we tried also to include the temperature into the migration probability between the two traps, but it was complexified. It was making the model more complex for not very interested. But the temperature was increasing the migration to the probability to migrate between the two traps faster. So I think, yeah, there is an effect like today, like if a fish was marked one day, if the temperature water was warm, it would, after when it was released, the smolt migrates faster than if the water was colder. So I think it does encapsulate a bit of this day-to-day variation. I don't know if I have answered you.
Yeah, I think you're saying it captures both the trend and the day-to-day. I think, yeah, but you're saying it does capture some of the day-to-day, so that's good. Do you want me to stop or do I ask one more question?
We have 30 minutes. No, 30 seconds.
30 seconds. Okay, one very quick question, easily dealt with in 30 seconds. What about taking this work and applying it to other sites? Do you see benefits of doing that for understanding climate change, space for time substitution or something like this?
Yeah, I think, yeah, it would be interesting to do this in other sites if the data allow it. But yeah, I think it would be very interesting to see if the things that we find in this analysis are the same as we think in other places, and more specifically because this is a southern population, and the southern population may respond very differently from the northern population, the more northern. For example, they survive in higher water temperatures and other more northern. So it would be very interesting to try to do this approach in other population. But first, we require the data to this. We have to have the necessary data to do this.
Yeah, and I don't have time to ask you about what the necessary data would be, so thanks for the time.
Well, thank you. I was like, oh, are there any more questions left now? But then, yeah, well, I have to say it was a great pleasure to read this. It's not only because it's Atlantic salmon, but also, I mean, the very high scientific quality of it was very nice to read. And you obviously have many different skills. You have done really good modeling work. You have done very good communication work. You're writing excellent English. You build it up excellently. Been very nice with all these little boxes, like, no, you have read this, and no, you will read this. It's very nice. And then you have these broad biological insights in the introduction. You have the Atlantic salmon biology. And also noted, not mentioned here before, artistic skills with all the graphics you have made. So it was very nice. So hope this is the last leg, and then you can lean back. And I think you should be really proud of yourself.
Thank you.
Yeah. Did you? I was wondering. I have more practical questions to begin with. And then, did you take part in any of the data collection?
Yeah, kind of. I went on the SCORF. So as you saw, the SCORF is in Brittany, and we are in the Basque Country, so it's a bit far, and I could not go every year. but I went during spring to see the data collection. And I helped a bit, but not too much. And yeah, so we are here, and this is here. So and so yeah, I participated in one data collection. It was very interesting to see what are the data, how they are collected, to see in. So, and I did also I a bit mature with electric fishing in September, because there are also electric fishing in September in the Scarf. So I participated last year to this.
So what was the challenges when you had to communicate with the people doing? They probably don't know your statistical models, and then the challenges with communicating with the field crew.
Yeah, I think the people that work there are, so they know a lot about the Scarf River and the salmon in general, but especially this salmon population. So I learned a lot from them, and I try to. They are collecting the data and a bit like in charge of the database. So they understand a bit what is like they understand the data and whatever. They don't understand the modeling, but they are interested in. So it's not. I think, yeah, it is. It is nice to work with them, and so yeah, I try to explain to vulgarize what was the data I needed and what information I was requiring. Was a necessity, but yeah. So
yeah. When you do complex models like you have been doing, you have made many assumptions of fish mortality, fish behavior, and so on. And it's sometimes difficult if you're not into the models in detail. It's always worrying what are, because you discussed a lot of these assumptions, but what are your biggest worries yourself? What's the assumptions you have made that you're most uncertain of? And how could you do field studies to get information about those biggest worries you have?
The assumption we've made in all the modeling approach, I think these are assumptions, for example, in the preparation process that we could not observe. So this is both a strength and a weakness of this type of modeling. So I would like to dig more into this preparation process, and for example, by using the individual data. So in September, there are electric fishing of the par, and the par are marked, and then some of them are captured as smolts in the trap. So we have individual data, and maybe with this individual data, we can inform all this preparation process. There are a lot of models that exist taking account the temperature in winter, etc. So we can also use this type of modeling and the individual data to improve this model and this mechanism, make this mechanism more certain. So this is my response, and just I think about something for the last question. I think what was also interesting with the technical team is that my modeling approach show some stuff that we did not know. For example, the fact that the capture probability was higher at the beginning of the period, as Jan showed. And for example, the hour at which the trap was checked had big influence on the data we collect. And before it was not considering as a big matter. And because of all this work on the data to understand all this observation process, this was very interesting because there were there were my results, there were their knowledge about the functioning of the trap, and we could communicate to make the protocol even like better and have better data. So it was very interesting. So it was just to make more complete the last answer.
Yeah, yeah. I was wondering about fish size. Did you consider to include that in the models in any way?
Yeah, where I consider it, but we wanted to put this into the model at the beginning, but it was quite hard to add this information with the way we model this, so we could not done this. But this is something also which is something that could be very interesting to know, if the bigger smolt migrate earlier, of what is exactly the link, because we, as we said, we often make the assumption that if the smolt migrate too early, it might be too small, but this is not exactly what we saw in the data. So it could be interesting to see, to better understand the effect of the size. And I think also what is interesting is more than the size is the growth, because there are two, like in some papers they found that the size has an impact, and in other papers they found that it was more like the growth rate that has an impact on the phenology. So maybe with the individual data we have from the smolt that were marked as par, and also all the smolt that are captured are measured. So maybe we can compare the growth and the size of the smolt, what's the effect on the phenology.
Yeah. I was also thinking, you assume you have no tag loss, no mortality between the traps. Is that one of your small or big worries?
The tag loss, we did not assume there were not. We took this into account thanks to probability to lose the mark. But yeah, for the mortality, this is one of my concerns, actually. We consider there were no mortality between the two traps. They are very close to each other, and this is often made in this double. This hypothesis is often made in this two trap experiments, because it is we cannot identify the mortality and the capture probability. So we do this hypothesis, but we also know that there are predators around the trap, and when they are stressed, they may be more risk for the smolt to be predated. So this is also something I would like we can check, like estimate the mortality between the two traps. But maybe it could be done with another experiment. For example, if we put like acoustic antenna, I don't know, to see if the predation is very important between the two traps. Maybe we can do something like this if we have money one day.
Yeah, I was also thinking like in relation to size, because this, it could be size select mortality, so that could relate to size.
Yeah.
You talked about avoiding traps, like they can swim back upstream, they might hear the trap, might be worried, that might also be size dependent.
Yeah.
And if you also have the interaction with size varying during the season, then you might have some complicated.
So we don't know if there is any effect of the size on the trap attractivity. This is not something we can, with the data we have, we can estimate. And for the effect of the mortality with the smolt, this is quite old protocols, and a lot of people are using pit tags. So I think it could be, but I think this is the risk is quite reduced, because we know that pit tag is quite okay for for the smolt if they are not too small.
Yeah. I was also thinking about radio tagging. Like now the radio tags are pretty small, so you could tag them and run around with your antennas and really track their behavior between and below or above the traps. Yeah. Also related to size, I think you use condition factor as a measure of size in chapter 4. And then I was wondering, because you have a rather surprising result with earlier pathology associated with a better condition, which could be a surprising result. But did you try, like condition factor is, that's their condition, like length versus weight? Did you try length and weight?
Yeah, I tried and I did not have a significant effect with 25% contil. But, and I had a significant effect with condition factor. So this is why I presented it. I'm not sure it means that the size does not have, that there is no effect of size. I'm not sure. I think to be sure of the size, the relation between the size and the timing of migration, we must do a better, include this factor into the model and do a better approach than a linear model with estimation, the results of our estimation. But yeah, the condition factor show a significant effect and not the size and not the weight on their own. Yeah.
As I was wondering if that might be related to not the size, but this multiplication process, like they grow in length when during the last part of this multiplication. So then the condition factor will be reduced because they get slimmer, which might be just an adaptation to the ocean migration.
Yeah.
So you could have that effect during the season, which is not the size effect, but it's the condition factor effect. Yeah. A couple, you also, one skill I forgot here was also the ability to relate it to management and discuss the management relevance for this. So I have a couple of management related questions in the end. Just like Jens said, we had a lot of questions when we were reading, both of us, we discussed this, and then, oh no, then you answered the question a couple of pages later. I was having this question, but what did you compare, like the data in your chapter 2 with just conventional Peterson-Mark recapture models, and then you did it. And then actually the results were not that different. Some years they're quite different, but overall not that different. Management related. So what was your advice to managers? Would they be well off with the Peterson-Mark recapture model if they just want to know the numbers, or would you recommend them to hire you and do a complex model?
So that's true, that's quite complex and it takes a lot of time and a lot of tries, so a lot of energy. So it can be hard to put all this modeling to make all this modeling just for an estimation of the number of smolt. And I don't think, I think mostly the Peterson is okay, but we saw that for some years this is not. So you must be conscious about this, that maybe your model is not is a bit misleading with this effect of the environment. So I think this is the optimal case is to do this modeling, but this is not possible practically for everyone. But for the phenological studies, as for phenology, we are looking at the number of smolt per days. I think this is much more important for the phenology, because if you think that you have very few smolts, but this is because of the capture process and not of the phenology, you are completely wrong in your analysis. So for an estimation of size, I think Peterson could be okay in most of the case. But for the phenology, I think you must be very careful with the capture probability.
Yeah. I was also thinking managers are interested in year-to-year data. So some years it can go really wrong.
Yeah.
That was the take-home message there. So then you started and ended in a way with climate change. So if climate change is in fact changing the timing of migrations, and it's non-adaptive for the salmon, then what can we do about it? We know it, we understand it, but then
so we can facilitate migration by opening the whales during the migration period. These are things that are made in management. So adapt the way we use the river, like with whales, with ecological facilities, etc. And we could also try to preserve the biodiversity of the population, of the small population, and hope that the smolt will adapt to this new condition. So I think, very practical management and other during the migration period, and other to maintain the population diversity.
Yeah, I think that was a very good ending. All managers out there listen, in times of climate change is even more important to keep the viable populations. And then last question, is there anything you would like to say or any question you would like to have that you haven't had so far that you would like to have?
I don't know.
If the answer is no, you're finished, you're off the hook.
My brain is too tired for that.
You feel you have said what you would like to say about your work now?
Yeah.
I think you have answered very well. Thank you very much.
Okay. So next we have to listen to your supervisor, who's going to speak on behalf of Mathieu.
Yeah.
Okay.
For sure. So thank you. So I'm the last. So finishing up, no question for you, Edel. We had plenty of time to discuss everything you presented. So also I will ask a question at the end, but it's an easy one. Not a scientific question. So yes, I do speak on behalf of Mathieu and myself, because we were two supervisors. And I take the opportunity here to thank Mathieu very much for his commitment in the supervision all the time and without any failures. So it was a great pleasure to co-supervise this work with Mathieu. So I just want to bring some information that might be useful for the committee to evaluate your work. The first thing is, I think it is, I think, at least obvious, but I can confirm that the modeling work you've been undertaking, it was not an easy one. So because of the nature of the data, and we've discussed this, and because of the amount of data you've been dealing with. So those CMR data, as you well demonstrated by the discussion on your presentation on your manuscript, they are clearly very noisy, and they were much more noisy than we were expecting at the beginning. So we discovered with you how noisy they were. And just because of the behavior of the smolts, because of the marking and the capture that interact with the behavior and create noise again, variability in the data. And on the top of this, because it's very difficult with those animals to maintain over 25 years, 3 months a year, a protocol very strictly, every day is the same. And so it's almost impossible to not to vary from day to day. And so I take also the opportunity, as you did, to really thank very much all the technical staff and students that over the 25 years of study have helped and make this possible. So really thank to them, even if many of them are not here or not working with us any longer, but it's really great thank for them. So despite those difficulties, Edel has managed to build nice models, flexible enough to accommodate this variability and to allow us to extract the information about the processes of ultimate interest we were dealing with. So that's what's the challenge. And this is also, I think we all realize that, but I can ensure that this is true. It's a lot of trial and errors. And it was difficult because of the amount of data, the quality of the data, but also because of the numerical tool we use for Bayesian inference, which are very nice, but especially Nimble and JAGS, we used a lot. But they are a source of a lot of frustration for students and supervisors as well sometimes. So every step forward is hardly gain for sure. And I want really to congratulate you, Edel, for your perseverance and for never being totally discouraged by what you were doing at some time. So really, you have a lot of merit. And the merit is even greater because at the same time you were doing this, you've also been teaching at university, 100 hours of teaching at university over the 3 years. Although I think you liked it, but it was not your choice, it was mandatory duty you had to accomplish on the top of your PhD. So that was another challenge added to this. And as you said, you have another constraint in France is that you have to stick to the 36 months. So at a point, you've got to do with what you have because you know more time to work. And I can ensure that Edel has gone up to the limit of what could be done in 36 months and start writing right to when it was right time to do it. So you overcame all these difficulties and you leave us with really founding work for us and with a lot of nice perspectives for studying and really understanding better what's going on with phenology of migration of salmonids, because we could apply this also to the adults. And you nicely helped us as well to describe and to define what were the perspective of future work we should undertake in the future. So thank you a lot for that. So besides your scientific contribution to the team and to science in general, you also had a significant contribution to our research community and research group. As you briefly said, you've organized, for instance, the no-pass workshop together with other PhD colleagues in the lab, but in 2023 in Pays Basque. So it was a very nice contribution, collective contribution. And also you helped to organize the other workshop, internal workshop for the lab, and also with other colleagues. So it was also a nice contribution to the life and the community life of the research unit. So I can say also that for sure, Edel has been very well appreciated within our lab. She's somebody who's easy to work with. She knows how to melt within a community and a research community, and how to contribute to the collective life of a community and research group. So that's really an excellent quality. So to conclude, yes, for sure, I want on behalf of Mathieu and myself, thank you very much and congratulate you warmly for the work you've done and all you've accomplished over those 3 years. So I finish with my little question. And my question is quite simple. It's professionally, what's next for you? Short term and middle term, what would you like to do?
Okay. Short term, that's easy. So in a month, I will be at the INRAE at Bordeaux, for still working on capture data, but on EEL this time. And new data and new cleaning, great. And so this is for 1 year. And after at least now, I want to continue in research. And more specifically, if I can, in migration studies, capture, and energy, and all this, all the things that I study in my thesis and former in my internship. And so after the postdoc at Bordeaux, I would like to maybe to go abroad, to do a postdoc abroad, and after maybe come back in France. We'll see.
Good. So thanks very much again.
Thank you.
Thank you, Etienne, and thank you, everybody, for this nice discussion. So now we will have to ask to the